So this afternoon, we're focusing on aquatic habitat and flow. And our presenter is going to be Dudley Reiser from R2 Resources. The session will go just like before. We'll have Dudley give an overview of the environmental baseline documents. And then we'll have a question and answer session between Dudley and the panel members. And then finally, we'll open it up to participation from the general audience. So let me introduce Dudley. Dr. Reiser is a fisheries scientist and president of R2 Resource Consultants. He has more than 34 years experience designing, implementing, and managing fisheries and aquatic ecology projects and habitat and in-stream flow assessments. His particular areas of expertise include fish ecology, both anadromous and resident species, habitat assessments and criteria development, endangered species evaluations, assessments of flow regulation on fish populations and habitats, fisheries habitat enhancement, fisheries engineering, in-stream flow studies, assessments of sedimentation impacts on aquatic ecosystem, and flushing flow studies related to sediment deposition. He's been involved in numerous technical studies related to water rights and defining in-stream flow needs for fish and aquatic habitats, as well as water resource development projects, including water supply and hydroelectric facilities. He has been leading the in-stream flow-related studies for the Pebble Project since 2008. He got a bachelor's degree in zoology from Miami of Ohio, a master's in water resources from the University of Wyoming, and his doctorate in forestry, wildlife, and range sciences with a major in fisheries resources from the University of Idaho. He has numerous publications in peer-reviewed journals, and he's authored chapters in 6 books. So, Dudley?
Is that the
Yeah, this
laser
right here.
All right. Thank you very much. It's always a challenge coming into an afternoon session after people have eaten to keep everybody engaged in a dialogue and discussion. So that's the challenge that I face. The other challenge is to present some what can be fairly complicated data and information out to you in a fashion that you can understand and sort of get a perspective on the types of studies that we've been doing that Pebble is engaged in over the past 5 years. The topic of flow and habitat is one that has been around for a long time. I've been involved in those types of studies for, as was mentioned, over 30 years or so. And I will say that right up front, there's always been this underlying discussion that sort of relates to what we talked about this morning of fish numbers, of trying to get some understanding of how does fish and fish habitat and flow, how are those all connected? And so I'm going to be talking to you this afternoon primarily about the latter two, the habitat and the flow perspectives. Those are the two elements that we've been looking at. I'll do that with a number of different elements too. We're going to be talking about physical habitat on the main stem, physical habitat in the off-channel areas that Mary Lou mentioned before, as well as water temperature, and as well as sediment transport and fluvial geomorphology aspects, of which I'm not a fluvial geomorphologist. So when I get to that, I'm a little bit outside of my realm of expertise. So those are the elements, and I'll touch on those again as we move forward here in the presentation. But getting us back again to the mine study area, Mary Lou went over these earlier, and it is difficult, as you say, Mary Lou, to see at this oblique angle. But we have 3 main watersheds, the North and South Fort Cook-Tule systems and Upper Tulare Creek right here. Now, each of these systems, as Mary Lou points out, from a study standpoint, we've gone ahead and stratified these into different reaches based upon various hydrologic, geologic attributes, just so we can begin to understand and organize the analysis that we undertook, both from the biological side of things as well as the habitat. Mary Lou showed these photos, so I don't need to dwell on them. The key thing with these photographs is, for those of you that have been to the sites and those of you that have not, generally what is characteristic of them, if you're first dropping into the systems, is sort of the uniformity of the habitat features that are there. And Mary Lou touched on this earlier. But if you look at the systems from essentially top to bottom in most cases, they're fairly uniform in their habitat types, what I would consider almost monotypic in their characteristics. And I mention that because oftentimes you get involved in in-stream flow studies or habitat characterization studies, particularly in the Northwest, for example, where you have large complexes of wood. You've got boulders, you've got undercut banks, you've got all kinds of attributes that are in the channels that serve to actually enhance or increase the amount of complexity that you have in these channels. These systems are fairly straightforward. There's not a whole lot of in-channel habitat. You don't see large complexes of wood in most of the areas. They're fairly open, as Mary Lou mentioned. And it's pretty characteristic of not only the North Fork, but also the South Fork, Cook Tule, and Upper Tulare as well. Now, each of these systems, from a watershed perspective, just to put a little bit of context for you, represents about 0.3% of the entire Bristol Bay watershed. So collectively, these three watersheds represent less than 1% of the Bristol Bay watershed in terms of area that we're dealing with. Mary Lou went through this figure, this table, which I'm going to have you study for 15 minutes. I'm just kidding. It basically is to point out that we've covered these types of studies this morning in terms of fish abundance, fish assemblages, and surveys. And I'm going to focus now on these latter part, these lower ones, the in-stream flow habitat studies. And you can see that years that they were studied, commencing back in 2004 and continuing through actually 2010. And in fact, there's continuing studies this year that were done on off-channel habitats. So there's a refinement, continuing out evaluations of certain aspects. The topics that I'll go through I mentioned already. The main stem channel flow habitat studies, I'll spend a fair amount of time on those. The off-channel flow habitat studies, I'll give you a snapshot of what we've done on that and where we're headed. Water temperature monitoring and modeling. Water temperature is a key element of fish ecology, so it's important to get an understanding of what the temperature regimes are in each of these systems. And then, of course, the fluvial geomorphology and spawning gravel characterization studies. The spawning habitats in these systems are very important. I view them as sort of the producers, the production zones of adult fish, and so understanding the quality of the spawning habitats is something that we've looked at. The components that I'll be covering for the main stem study first include the fundamental tools that we've used. And when I say tools, that's really, I want to spend just a second in talking about that. What we are trying to do with the baseline characterization from the in-stream flow perspective is to develop a set of tools, analytical tools that can be used not only to characterize baseline conditions, baseline characteristics, but also to look at potential effects of mine operations, the mine footprint, when you get down to that point. So that we've got the framework for being able to look at different scenarios, different flow conditions that might occur with and without the mine. That's really the goal of the work that we've been involved in, developing those set of tools. Part of the in-stream flow study relates to habitat suitability curve development. I'll touch on that. And then what are these things called flow habitat relationships? What do they look like? Give you an estimate of that. And then pulling it all together on the main stem, estimating habitat quantities under different flows. Off-channel habitat, elements, I'll talk about the different habitat types, off-channel habitat types, and where we are right now with carrying that forward. Water temperature monitoring, I'll touch on that, the baseline thermal conditions, and then discuss a little bit about SynTemp, synthetic temperature model, stream network temperature model development, where we're going to be able to look at how temperatures might be influenced under different flow conditions. So that's the goal of the SynTemp modeling. And then fluvial geomorphology. One might ask, how might the project, how might the mine development actually affect flow conditions in these different systems? Well, there might be no change in different locations. Maybe absolutely no effect that you would see from a hydrology standpoint. But there could be temporal changes, the frequency and timing of certain flow metrics. The peak flows, for example, may change. The base flows could potentially change. There could be spatial changes in hydrology, magnitude, the rate of change. A lot of these flow-related effects, if you've been involved in hydro projects, these are the types of things that one might associate with hydro projects, but they also can occur with other types of water resource development projects, including mining. So that's what we were trying to focus in on in the development of our tools. In developing the approach that we used and selecting the methods that we applied, there were a number of considerations that went into that, including, we want to be able to look and evaluate, since we're dealing with a fairly wide geographic area, and each of these systems is over 30 miles long in length. We wanted to develop a system that would allow us to evaluate when, where, and to what extent the changes in flows might influence different habitats at different locations in the stream. So this fundamental baseline characterization aspect is one of the key things that we were looking at. What's out there right now, under what different types of conditions, under natural flow regimes. And you'll see that we talk a little bit about wet year, dry year, normal year conditions. So we want to look at the full range of habitat conditions that occur under different flow conditions. We needed to consider the local hydrologic conditions, and for this we relied heavily upon the work that Knight Piesold has done, that some of you probably listened to last week on the hydrology. So we worked closely with their hydrologists in conducting this and using information that they provided. And we wanted to be able to, like I said, look at habitats locally, longitudinally, and cumulatively. And then we wanted it to be adaptable to where we could evaluate project impacts. Now, the primary approach, when you get into these types of studies, if you've worked in the in-stream flow arena, you know that there's not any one particular method. There's a variety of methods, in fact, that could be applied. And not one single method is going to address all of the flow-related effects that you'll incur. In this case, looking at the conditions that we were dealing with, we were interested in a methodology that could evaluate flow effects incrementally. And the primary approach that we picked for that is the PFSIM, or the in-stream flow incremental methodology, to do that. What I mean by incrementally is this is a XY graph with flow on the x-axis and habitat on the y-axis. And so when you apply a PFSIM type of analysis, you can actually plot out habitats over different flow conditions. So you can begin to see relationships for a given species and life stage at a given location, how habitats are changed or how flow is influencing those habitats. That was the primary methodology that we've used, but of course it's not the only one. As I'll mention, we have the temperature monitor models, fluvial geomorphology, we're looking at off-channel, And of course there's considerations too with groundwater and upwelling conditions too that are important in terms of defining fish habitats. From a purely sort of mechanistic perspective, PHAB-SIM, Physical Habitat Simulation, that's what the acronym stands for, includes a number of elements that we'll step through here quickly. They include a field component where you're collecting the data. This just shows a cross-section of a stream where measurements are taken across the channel. It includes habitat suitability criteria that feed into the models, that bring in to bear the biological utilization, if you want to consider it preferences, of fish and life stages to these attributes that you're dealing with. Habitat mapping, I should have actually started down here. Habitat mapping is an important part. We need to be able to determine for each of these systems the composition longitudinally of different habitat types. That's important so that we make sure in the transects, the measurement, the field measurements that we're collecting, that we're selecting representative habitat features that we can use then to extrapolate to other areas that are unmeasured. So habitat mapping is a very important part. And then on the right-hand side then it just shows, again, these relationships, habitat flow relationships that are derived from here. Beginning with the habitat mapping, Mary Lou showed a figure similar to this, and we've colored this now just to show you different habitat types that we were able to map with aerial photography, orthophotos. We've actually divided the river now into literally hundreds and hundreds of habitat types that we've been able to delineate that have factored into our analysis. On all three of them, the North Fork, South Fork, and Upper Tulare. This is just a blow-up of the cross-section profile that I mentioned before, showed you before, showing you generally how these measurements are made and how you develop the habitat flow relationships. You start with field data that's collected, and they're collected at different transects across the channel. I'll talk about the numbers of transects that we've used in a minute. But basically, you line yourself up. You're going to put in the simple form, put a tape measure across the channel, and you take a set of verticals where you're measuring depth, velocity, and substrate composition, some cases cover, that you find across that transect. So that's representing combinations of depth, velocity, and substrate serve to define habitat, fish habitat, for some species, not all species. Some species respond differently to conditions, and they don't fit, for example, velocities. You know, sockeye and chum are good examples of species that in spawning selection, they don't always zoom in on flowing water, velocity movement. It's more of an upwelling type of a situation. Well, what you're trying to do is establish a hydraulic model or develop a hydraulic model. So we will select 3 flow measurements. We will actually take 3 different flow measurements that we're trying to capture what we'd call a low flow, a mid flow, and a relatively high flow. And in selecting those points of measurement, we're trying to select those points that we believe will capture the shapes of that habitat flow relationship when we go through our hydraulic modeling. That'll capture that range of flows that will allow us to actually define the habitat flow relationships. We don't go out in the winter, if that's the question. We have to have flowing water, and we'll do it in a summer period where there's a low flow period, and a springtime period where there's a higher flow, and then we'll capture a intermediate flow somewhere in between those two. Excuse me? We've got gauges that we have set up that Knight Piesold is monitoring that can tell. There's an extensive network of stream flow gauges that are present.
Yes,
sure.
I guess two things. Is, number one, if we could try to hold most, if not all, questions until the public comment section. And then number two, if you do get a question that you need to answer, could you repeat it for
Sure.
the...
Yes, absolutely.
Thanks.
You bet. Okay, so this is the general procedure that is used in collecting the habitat data that goes into the models. Now, in 2004 to 2007, this is prior to R2's involvement on this project, we completed, just like Mary Lou did with the fish data, an extensive QA/QC of all of the data sets that were present at that time. There had been a total of 92 transects established and data collected on those 92 transects in these different systems. 2008, after reviewing those data, we decided that we would add additional transects, what we called intensive surveys. And by that, I mean the 92 transects that were used in these studies were spread out in different locations throughout each of the river systems, trying to capture habitat types within representative areas. In 2008, we concentrated, after completing aerial recons and getting familiar with the sites, we added 46 transects in areas that we thought were especially biologically important: spawning habitat, juvenile rearing habitat. So we focused our efforts in 2008 on intensive surveys, 46 transects with a total of 138 transects being measured then. Part of our QA/QC process, when you go through this information and you get your hydraulic modelers developing models for use here, is they're going to be looking for things that are anomalous, that might be problematic in terms of calibrating the models. In doing that, we identified 21 transects that warranted additional measurement. And so we've gone back now and resampled those 21 transects. The EBD that you have, that you've all been reviewing, is therefore based upon 117 transects. The newer information, the refinement of that, incorporates the rest of the transects into it. So that is a difference between what you've reviewed and where we are right now. As I say, things are continuing in terms of evaluations and further refinement. So of the distribution of these transects, we got 117 transects, 27 of which are in the North Fork Coktuli, 3 transects in one of the tributaries to the system, 37 in the South Fork Coktuli main stem, 6 in a tributary stream, etc. You can read these. 35 in Upper Tulare Creek and 5 in Upper Tulare Creek tributary, which is the tributary that Mary Lou mentioned as receiving waters from South Fork Coktuli, the 25 CFS on an almost continuous basis. These next series of figures display then the distribution of transects that we have first in the North Fork, and just a point here, here's the ore body, here's the North Fork coming up, and you can see that we did concentrate the transects in this portion of the stream in areas that we knew would be most sensitive to flow reductions, as well as areas that we knew received the most use from a fish use standpoint. In other words, the lion's share of the spawning habitats are contained within those reaches, within that region. Likewise, in the South Fork, the same situation. Majority of the transects were located in SFK-B and SFK-A. There are a few transects, though, that we've got in SFK-C. SFK-C is that reach that we talked about earlier as going temporarily intermittent at certain times of the year. So we've got some transects, and we're able to evaluate habitat flow relationships even within portions of that reach, SFKC. And then here's the relationships or the transect locations for Upper Tulare Creek. Now, real quick, because this could really put people to sleep, and I know there's struggling going on right now. I can tell it. I have the vantage point here, so I can see what's happening. So I need to liven it up here, I think, is the key thing. So there's a calibration process that you go through. We use hydraulic engineers for the whole process on our staff. There are companies that will use biologists for doing this, very knowledgeable biologists to do calibrations. We opt for always going with hydraulic engineers that understand the mechanics of stream flow and hydraulics in systems. So when you're calibrating, they make informed decisions in terms of what they're doing. One element that Mary Lou sort of touched on, but we didn't get into great detail, is periodicity. Periodicity, and I'll talk a little bit about it, is very important for the analysis that we completed because it defines the timing and the life stage of when different species are using these different watersheds. So it's a monthly periodicity that we define. And also, I'll touch on that in a minute, suitability criteria. There's a myriad of ways of developing suitability criteria. Each one of them has its critics. There's no right or wrong way of doing it. I think the way that you do it needs to be documented and evaluated, and that's what we've been doing, collecting data so that we can develop site-specific criteria. And then just calculating the WUAs for individual flows. This is a periodicity chart. It should look familiar to you from the EBD. And these red areas just represent areas of intense, the greatest activity for that, and they're also based upon observations. So not only did we get information on timing, on numbers from the aerial surveys, but we also got information on timing of when fish are entering the system, when do they migrate into the system, the adults anyway, and when are they spawning. So when we have that information, we're able to bring that into the periodicity data and base that on the existing or on data that's specific to those watersheds. Suitability criteria curves, important. These are the building blocks for what we define as habitat in the systems. This gives you an example of frequency distribution of observations for coho spawning velocities. So these represent 98 observations, and of those 98 observations, you can see the distribution of those observations in terms of the velocities associated with the fish that we observed. So it gives you an idea of what velocities they're really homing in on, zeroing in on. There's a range that you can see here. This is a sort of a frequency distribution, and then this would be the type of curve set that might come out of that in terms of what's applied in the computer models. And what happens here in the suitability, or in the suitability criteria, is that this then becomes probability of 1, meaning that's the best habitat characteristic, the best attribute, and we've extended that over to, in this case, around 2.4, 2.5 feet per second. The way that WUA is calculated, weighted usable area, is calculated for every one of the cells that we've measured across the channel. It has a combination of depth, velocity, and substrate. The weighted usable area in that particular cell is the product of the individual suitability criteria values that are multiplied together. So there's a velocity suitability index value, a depth suitability index value, and a substrate index value. Multiply those three together, and that calculates your weighted usable area for a given location under a given flow condition. And then, of course, that's rolled up with a computer program, and you're able to generate habitat flow relationships. In the case of this project, there was a combination. You start off with what's known in the literature. There's been lots of studies done defining habitat utilization for Chinook, coho, chum, pink salmon, sockeye salmon. So you start with that information. But then we build on that by going out and collecting suitability data by snorkeling, snorkeling the streams, observing fish, identifying the size class, as Mary Lou said, the bins, and then coming back, you flag the locations of where you've observed those fish and come back and take a measurement of depth and velocity. And so that becomes your point, your measurement of habitat utilization for that particular species and life stage. For reds, for spawning reds, it's a lot simpler. You know, you want to be able to survey and identify fish on the reds, and then you go out and take a depth and velocity measurement and record that. Gives you an idea of the numbers of observations that we've made, over 1,300, and you can see that this, you know, we broke it out by the two different companies, HDR and R2, and here's the total at this point up here. And you can see that not every species or life stage is equally represented. We've got, for example, plenty of sockeye spawning observations. For over 400, we have, you know, less than 8 observations for rainbow trout spawning. And that's a function of rainbow trout spawn in the spring, high flows. It's a real problem getting out in the streams and actually identifying locations. So we're going to have to use a combination of some of measurements like this, as well as then going in and looking at the literature values and seeing what's realistic. Now, that sort of is the building blocks behind the PAB-SIM model, the main stem models that I've mentioned. So we've got, you roll that all together, we've developed models for each of these systems, and you're able then to generate different habitat flow relationships for different species and life stage combinations. So here we've got Chinook spawning. This is for an average water year type. And when I say spawning, what we've got here, that relates to the green figure here. That's why this value here, this statement of average year comes into play. This figure is representing the habitat flow relationship that we've derived for this location. This happened to be in 2005, and this is SFK, so it's the South Fork. It's in a run habitat, and it's the 7th run that was measured in that reach, in that section. So that's the nomenclature and how this is laid out. So at that location, we've defined this relationship, but that in and of itself doesn't tell you a lot, other than, you know, if you had the full range of these flows at that location, this is the type of curve that you'd end up with. So you have to bring in hydrology into the picture to understand what does this telling you. So in an average year, when we look at Chinook spawning, we go to our periodicity table and we pick out the months of spawning. So it'd be July, August. And then we query the hydrology database and figure out what the average flow is over that spawning period, those 2-month periods. And then we come down to our curve and say, well, here's the average flow. It's a little bit less than 100, and that's the amount of habitat that you'd have at that location under an average water year condition during the spawning period of Chinook. We've done this for each of the species and each of the life stages. In the case of juvenile, you know, it's longer than 2-month period. Juvenile rearing can go on 12 months of the year. So we will then make sure that our analysis considers the hydrology for the entire period of year, the entire year, in making that analysis. We used the 68 years of monthly flow data. Knight Piesel described this, I'm sure, last week. 30 hydrology sites. We're able to actually longitudinally, because of the network of hydrology gauges that we have in these systems, we can adjust longitudinally the flow conditions at any point down the river system based upon the hydrology gauges as well as the groundwater influences that we know through groundwater modeling that have occurred. So we're able to adjust and understand longitudinally the hydrology as it moves downstream, and if we've got a transect that's down in SFK-A, we can adjust and make sure that the hydrology that's there under an average year condition is consistent with that, and we apply that instead of the hydrology that would occur in SFK-B, for example. One of the big questions that always comes up with in-stream flow studies when you're dealing with whether it's hydro projects or any water resource development is how do you take the information that you've gathered at certain locations and expand that to the rest of the river. You've got to make some assumptions about that. There are ways that it's been done. There's multiple ways that it's been done. And the way that we applied this in the EBD, I'll describe. We have different what we call meso-habitat units, basically the pool, riffle, run conditions that we're able to delineate in different locations as we go downstream. By doing that, you get a sense here, there's somewhere in the range of over 75 units in SFK or NFKA, 152 in NFKC. You can see the range of habitat units that we've been able to delineate as part of this process. Now, in the approach that's often used in in-stream flow studies is you weight these transects. You weight that information that you gather at a given location, for example, TRA, which is a run habitat. Halfway, that transect then is weighted halfway to the next transect that's measured. So you weight it halfway here, and then this transect would be weighted halfway downstream or upstream, whichever way you're defining this. And that is a pretty standard way that weighting is taking place, so that you can take these weighted usable area versus flow relationships, weighted usable area being square feet per 1,000 linear feet of stream, and translate that into area. Because you're really interested in weighted usable area versus flow relationships here for the whole reach. And so you bring in that component of length and what's the weighting factor that should be applied to these different lengths. So the problem that can come up, or one may say is a problem here, is that if you've mapped out different habitat types in between here, and we know that they occur, then you're essentially missing those. You're not really capturing the variability, if you will, that occurs because of those different habitat features. You're simply weighting this run habitat halfway down to this transect here, and this transect halfway up to this one. And you're missing all the rest of these transects. And that's the method that was applied in the EBD, just so you're aware. This is how we've looked at it. Now, we've subsequently thought about this and have come up with what we consider to be a more refined approach. This is not in the EBD, but it basically, we're normalizing the known transect information to bankful width and the annual Chinook spawning flows. This is just an example. We've done this for each of the life stages. And by doing that, we can normalize it, we can simulate, and we can actually identify or create a weighted usable area versus flow relationship for each of the habitat types, unmeasured habitat types, based upon a combination of weighting factors and normalization. So that in the end, instead of relying on just weighting one transect halfway down to the next, we now have the ability to actually develop individual weighted usable area versus flow relationships for every habitat unit type in the system. And we've conducted our analysis beyond what we've done in the EBD using that approach. I consider it a much more refinement to the process that we applied in the EBD, and I think it's I think it's a creative and innovative approach that has application outside of just this work that we're doing here. Now, what do you do with this information in the end and what's presented in the EBD? See, how am I doing for time? Doing okay? I'm about an eighth done. I'm just kidding. Just kidding. No, I wouldn't do that to anybody. So yeah, what do you do with this information? Well, there's a number of metrics that we've developed and have applied and listed in the EBD. And this weighted usable area, which is simply the habitat area per 1,000 feet of stream. You can also look at it as weighted usable weighted usable width, and begin to understand how habitats change longitudinally. And then there's cumulative habitat, so you can begin to see on a scale, on a longitudinal scale, where are you gaining the most habitat in the stream system. That's one of the reasons that we went to that approach, is do we actually see points where there's blips that says there's a lot of habitat features going on here. And then, of course, you can go into the sort of a normal approach, which is time series analysis and habitat duration analysis, which is often used down the road when you start thinking about project effects, project impacts. You can look at your time series analysis and with project versus without project, and you begin to get some idea of trade-offs between, what are you losing, what are you gaining under different flow conditions. An example of this, I'll just go through this very quickly. One of the linkages back to this analysis, of course, is hydrology. And I mentioned, in this case, for Chinook spawning, we're dealing with 2 months. This just shows you the wet, dry, and normal year patterns that we identified, our hydrologists identified, that were used in the analysis. This was done for every life stage, and the periodicities, as I mentioned before, the same thing. We had to go through this process of identifying the hydrology set that we're going to run through the analysis. Part of what you can do with the habitat duration, this is a schematic that just shows if you start with flow, habitat versus flow relationships, you can bring that together with your habitat, your hydrology. This is just flow over time. Bring that with your habitat flow relationships, and you can develop this habitat time series, which is habitat over time. And that's useful, as I say, in looking at the baseline conditions, average, dry, wet years, and then we can look at that in terms of project effects. Same thing with habitat duration. You can look at the flow duration curves, tie that in with your habitat flow relationships, and come up with habitat duration. Just another tool that can be applied in our analysis. It's an example of weighted usable width longitudinally. You can see that as you progress from upstream to downstream, you've got blips of habitat. This is, like I mentioned, useful in identifying where are you getting the large hits of habitat in different conditions, so very useful as well. And then cumulative habitat. You see this is listed as baseline. This could also be run and evaluated with different projects, so you get a sense of what's happening here. Can also bring forward then, just in a summary fashion, habitat amounts. terms of acreage. I think there was some questions this morning about the areas, habitat area, and how much spawning habitat do we have in these stream systems. PFSIM is one tool to get at that question. It's not the only tool, but it does provide you information on defining how much spawning habitat you might have in for Chinook or coho for the entire system, or at any different location that you have these transects, and you can tease out that information longitudinally. So it provides that ability to actually summarize and provide estimates of the amount of spawning habitat by species, by life stage. This is just an example of Chinook juvenile. And then you can tie it all together, bring all three streams together, which we did in the EBD, and look at Chinook habitat, and you can look at coho, spawning juvenile. We didn't even present fry habitats here as well. So that sort of takes us through the main stem work that we're doing. And there's a lot there. There's a lot of information that we've presented. I won't be going into as much detail for the remainder of the discussion this afternoon, thank goodness. But I just wanted to take that time to let you know that there's been lots of very detailed work done in developing these models that we can move forward then in evaluating potential impacts. Now, Mary Lou touched on off-channel habitats this morning, and one of the things that we've been interested in is, so you have this model that we've developed for the main stem, and we feel like we've got a pretty good idea of how habitats in the main stem, main channel change with flows. Let's make that assumption that we do. The question is that the entire picture for fish habitat? And certainly we all would, in the room here today, would say, no, that's not the answer. That's part of the answer. Coho juvenile rearing, for example, we know is concentrated in in off-channel areas. There'd probably be people, agencies, scientists, knowledgeable scientists that would say coho really use off-channel areas. You don't really need to even think about them in the main stem, for example. That's a possibility. So one of the things is to look at these off-channel habitat areas. And we have, as Mary Lou pointed out. We've done this, I would say, in two stages. We've completed an initial surveys that were done and are reported in the EBD that serve to identify the areas that we've classified as off-channel habitat types. The beaver ponds, the alcoves, the cutoff ponds, those types of habitat features. The key issue that we're now looking at is this interconnectivity relative to the main stem flow. And that is, how do these habitats function relative to the flow conditions in the main stem? Some of them are likely independent of the main stem flows because they might be groundwater fed. Some of them are connected. And for those that are connected, we want to understand what those relationships are, so that if you increase the flows in the main stem or decrease the flows in the main stem, what does that do to your total area that you've got in off-channel habitats? So that's what we've been embarking on this last year. These just give you some shots of different habitat types in these off-channel areas. These are side channels. We've defined these in the EBD, backwater slough areas. It noted here water surface elevation is strongly correlated with the main stem in these backwater slough areas. Isolated ponds. We found in some of the studies where we put in pressure transducers into these different types that, in this case, isolated ponds track pretty well with the water surface elevation of the main stem. Groundwater on some of these other habitat types seems to be a major influence, and there's not that great of a relationship. Overflow channels fed by groundwater. They're connected to the main stem and accessible to fish even at summer low flows. So there's certain types of off-channel habitats that may be, even over the full range of your main stem flow conditions, are still remain connected. And we want to be able to identify those. And then you've got your beaver complexes that do maintain a connection, access for fish. This is a good spot to talk briefly about. There was a comment this morning about beaver activity, and I'm not sure if I can't remember what the issue was, what the question was, whether there was a plan to remove beaver. Yeah. Was that the question? If I could, just for a second, sure, to capture that one, because I didn't want to lose it.
Yeah, I just wanted to know from your vantage point how important beaver habitat was for salmon populations of your particular rearing stand, because I don't think this microphone's on, but because in comments to the EPA on the watershed assessment, I know these are two separate processes, but
right,
in comments to the EPA on the watershed assessment by Northern Dynasty and Pebble Partnerships, there are discussions, albeit brief, discussions of removing temporary and permanent barriers, such as beaver dams, to improve habitat as a mitigation option. So if you're going to lose habitat because of a mine, then you're going to improve habitat elsewhere. Removing beaver dams was one of the proposals, and that seems counterintuitive given your own data. So that was the context.
Right. No, that's a good question. That goes back to, I date myself a little bit, but you can think back on the conventional wisdom when I was a grad student coming up about removing wood from the stream. In the lower 48 anyway, there was a whole push of just removing these debris jams, and we know better now. We know that that's an important part. We put wood back in now in a lot of stream systems. So from our perspective, the beaver activity is part of the natural ecosystem that's there. And I think we've identified that there are important elements to this. To think that we would have a major program and going in and removing beaver would be counterintuitive, and it would be something that would, only in extreme circumstances may that be something that would be considered. But for the most part, we consider beaver as being part of the ecosystem, and would not embark on something like that. Yeah. These just show Mary Lou showed these, so I'll skip through these pretty quickly, and I am going to step it up a little bit because I'm already pushing 1 o'clock, or is it 2 o'clock? Yes, at 2 o'clock. So these are the off-channel habitats that have been identified. The next steps that we're going to be doing with this, as I mentioned, are going to be largely a GIS and field ground truthing exercise, but tying these off-channel areas into the hydraulic and hydrologic models that we have on the main stem, with the goal that we can dial in, for a given flow, we should be able to, for the North Fork, South Fork, Upper Tulare, say, well, that flow provides generally this amount of acreage of off-channel habitats, and we can even try to get into defining what constitutes habitat, the water depths, et cetera. But that's the goal, and that's where we're headed with the off-channel work. With water temperature, I can go through this pretty quickly, but I do want to stress that water temperature is one of the main drivers in salmon ecology. It controls a lot of the physiological behaviors of fish, the reproductive components of fish, when fish spawn, when do they migrate. A lot of these things are all linked to temperature. So understanding temperature in these systems is very important. There's a wide array of temperature data that are contained in the EBD. I'm not going to attempt to go through these data in any detail other than just show you snippets at selected sites. This is 2007. And then this is giving you just sort of a spatial and temporal evaluation, or a snapshot of where some of these monitors are located by river kilometer. And this is in the North Fork. And there's a complement of these in the South Fork as well as in Upper Tulare as well. And you'll notice the red lines there indicating tributary water temperatures. Those are important. Those data are important for calibrating the SynTemp models that have been developed. Quickly, here's a snapshot over time. This is at river kilometer 13.7 on the North Fork, and you can see some of these are just showing you the range, the maximum and the minimum temperature range that we would see. This is in 2007, so we've got data from other years. But it shows you that there's a lot of information like this that we've got over time. And then we also are able to look at different flow conditions longitudinally. And how does the river respond in different locations under a given set of flows? This is on August 21st. And you can see the changes as you go downstream, little blips here, that might be a groundwater point, as you're looking at your thermal profiles. And you can see how this changes over time. We're in September now, and here we are in October. And, eventually it just pretty much the maximum, minimum. There's not much of a range between the two. And here's South Fork, same type of information presented, and then here's the range in the South Fork at river kilometer 29.3. A little cooler system at this location anyway than what I showed on the North Fork. And again, you can see the longitudinal changes, again, a big groundwater signature here occurring. And see how that changes over time. Likewise, Upper Tulare. I'm going to step through these very quickly. But this type of information, there's a lot of this type of information in the EBD. So I'm just pulling out a few examples here of this. And then final one there. Now the temperature network and the temperature model then relies on a variety of data, hydrologic data, temperature data, meteorological, riparian vegetation data, etc. There's a suite of parameters that you need in order to develop these models. This is just some of the meteorological data that was used in pulling this together. And the river valley morphology data, that feeds into it as well, longitudinal profiles. Each one of these parameters has a physical value and a need that's brought together into the SynTemp model. And so these are the types of attributes that are factored into the temperature model. Now we do have then models developed for SFK, NFK, and UT. And the big point, second bullet here, well, both of these bullets are important. We can, with these models, begin to understand how flow changes might be influencing water temperatures at different locations down the river. Why is that important? Biologically, understanding how temperatures change will help us understand whether there's likely going to be any effects on egg incubation. Is the incubation period going to be longer, shorter? Emergence timing, growth rates of fish, overall productivity of the system is all linked in with water temperature. So it'll be a powerful tool that can be used in moving forward. One thing, too, to point out is this intergravel temperatures. And there was mention of the hypereic zone, which studies have shown is very important for fish and for the incubation environment and for invertebrate community structure. And if you go onto the river and you're walking the river system, you will find areas where you actually have visual evidence of groundwater upwelling. One of the studies that we've looked at, this was actually done by HDR, completed some intergravel temperature monitoring during 1 year. And we were able to, with those data, evaluate and detect certain locations where, we have, for example, in the case of the figure on the left, we are at river mile 20.6 It's about 7/10 of a mile downstream from tributary 190 on the South Fork. And you can see that at that location, water temperatures actually went to 0 in that location. So whether that's a function of complete dewatering, that we talked about the ephemeral reach. If you go downstream about 1.1 miles from tributary 190, you see that at 30 centimeters deep, you've got water temperatures that are above freezing. So there's clearly an influence that you can detect of groundwater and the importance of groundwater. And that's just a part of these. Each one of these systems has got that type of a interaction at different locations. And, Hugh McCready and Rod Smith last week, I'm not sure which person was here last week, but they would have given you a good overview of the groundwater modeling that's been done that will help, help us all understand what these upwelling areas are, how they might ultimately be influenced under different conditions. So that's a key thing that we want to bring into the fish habitat part of this as well. Last but not least is the channel forming flows. And the key here is we understand that what we're looking at right now, when we do our main stem work, we are looking at a set of conditions that have been provided and created through literally thousands of years of the hydrologic scheme and the regime that's present there right now. So that channel form, the channel geometry that we have there, is reflective of those conditions. Now, if we bring the project into play and we change the flow conditions in there, you have to be able to say, or evaluate what will those changes be, potential changes be, in the future. So that was in part one of the reasons for looking at channel morphology and channel forming flows and understanding what those relationships are, what flows are needed to create the habitats, to sustain the quality of those habitats. So there's been a program involved in that. Geomorphology study sites. Again, there's an appendix that covers this. I wanted to make sure it was included just so you're aware of it. There's even studies that we're looking at, if the next slide will come up, at this particle mobility study that was done, actually radio tagging rocks in the North Fork, Koktuli, and trying to get an understanding of particular flow conditions that would mobilize the bed as one way of understanding those habitat forming flows. So there's a whole bunch of regime theory that is out there. If you talk to scientists involved in those types of studies, you'll understand that channel forming flows, is it the 2-year or 10-year flood event, peak flow that's causing those flows? We'll be looking at those types of flow, those hydrologic events, and see what we think the effects might be on channels, on channel characteristics. This is just a schematic to show you that if you assume that there's a relationship between hydrologic metric and a channel forming flow, you have some type of a relationship here. And this is conceptual, so this is not anything that's based upon any data. But if you assume that there's a hydrologic metric here that's related to channel forming flows, and you've got this relationship, you can see that if you change that, here's the project effect, you can come down and move that, sort of move that through the sequence here. Here we've got channel size versus channel forming flow, which is, there would be that relationship. And then you can bring that all the way up to habitat quantity versus channel size. So that is just conceptually how we're thinking of addressing this question of changes, long-term potential changes in channel form and how that might affect fish habitat. We're not only looking at the short-term effects, potential effects based upon the main stem habitat flow, but looking at it from a longer horizon of what might happen in the future if there are changes in the flows. This is just there's be a short-term habitat response, as I mentioned, versus a long-term. So that's what we're looking at. And this is just a figure that I'm going to skip over, as well as this. This is just an example of bank full reduction by 50%. And one of the final thing here, and I'll get done here in about 2 or 3 minutes, is the spawning habitat characterization. So the question that we had was, in walking out there and looking at the habitats, we believe that the habitat spawning gravel quality is very good. But the hydrologic regime out there is interesting in that you have, well, here's the spawning gravel sites. Well, let me go back here. You have 2 high flow events occurring. So you've got a period of time when fish are spawning in the summertime and the fall that's followed by the fall rains. So that has the potential of mobilizing sediments and bringing those sediments into those newly constructed redds, if you will. And sort of the question was, well, is that, what's the quality of those gravels prior to spawning versus after that initial set of flows, the freshets that come in. And so it was useful, what we're trying to do is figure out when is the best time to actually characterize the spawning gravel quality. Do you do it beforehand, or should you wait until afterwards? And so that's what the focus of this study was on. And we sampled 3 different locations, had replicate samples at each of the sites. This is the hydrograph that you may have seen from other studies where I just pointed out here's your spawning period that extends all the way up in case of coho, for example, all the way into November. So you've got an incubation period that is occurring, as a subset of the spawning, and extends all the way out into potentially April. And here's your typical hydrograph. So you got high flows coming in here that's bringing in sediments. And so we did a pre and a post sampling analysis where we collected gravel samples, 6 replicates per site pre-spawning in these different areas. And then we collected 3 replicates per site post-spawning and post-fall freshets, and made a comparison between those 2 concentrations. So there's a variety of metrics that we used in qualifying what the gravel quantity or the gravel quality is. It's just an example of where the sample locations are. In the October sampling, we actually did find alevins, so we knew that we were in proximity to some of the redds. We tried to avoid this. There were occasions where we would find a couple of alevins in there, but we knew that our sampling technique was capturing these areas that were being used by spawning, used for spawning. The results were this is summarized here. If you look at the solid bullet versus the open bullet, one is August sampling and one is October. You can see the difference. Some differences occurred between the different locations and different times, but generally there was no statistically significant difference between those 2 samplings. And importantly, the metrics that we're using here, this percent fines less than 0.84, all of these samples are well below that, below this threshold here that's been looked at, indicating 50% survival. Same thing with other metrics that we looked at. This percent fines less than 6.4. There's a couple of other metrics that are often used, geometric mean diameter, and the Friedel index, another one that's been used. And invariably, the gravel quality that we found on either pre and post is very good gravel quality. Spawning gravels out in these 3 systems I'd consider them to be excellent quality. So, I covered a lot of ground, and I think I am rapidly approaching the point of saying that's all, folks. So, I will. Thank you.
Right, thanks, Dudley.
Yeah.
All right, so I think this is probably a good time to take a break. We'll come back with panel interaction with Dudley, and I think Mike Stone is going to have some points he wants to make before we bring everybody up there. So let's reconvene at 2:30.
Sounds great. All right.
Thanks.